a human, a chicken, a turtle, a salamander, and a fish. In their adult forms, these 5 animals look nothing alike, adapted for entirely different environments and ways of life. Looking at them side by side, it is difficult to identify a physical trait that could possibly link these species together. But if we look past their specialized adult features and return to their earliest moments of development, the physical differences begin to fade. At this primary embryonic stage, these 5 organisms are virtually indistinguishable. They share the same basic body plan and identical anatomical structures. This raises a fundamental question. Why does the beginning of human life mirror the early development of a fish or a salamander? To understand the connection, we can isolate the human embryo and examine its initial anatomy. In the neck, the embryo develops pharyngeal gill slits, the exact structures that become gills in fish. While humans eventually rely on lungs, these aquatic features always appear early in gestation. Lower down, the embryo possesses a distinct tail supported by V-shaped myotome muscles. This tail is reabsorbed before birth, but its temporary presence is a physical echo of ancient aquatic ancestors who used these muscles for propulsion. We see a similar pattern inside the embryo's earliest forming heart. Initially, it is a simple pump with 1 atrium and 1 ventricle. This 2-chambered arrangement perfectly matches a fish's heart, existing before differentiating into a complex 4-chambered mammalian organ. The human body begins its development by following these ancient aquatic blueprints, revealing a deep biological connection to the water. In the 19th century, German scientist Ernst Haeckel recognized that these physical patterns were not coincidental. He proposed the biogenetic law, famously summarized by the phrase, ontogeny recapitulates phylogeny. Essentially, ontogeny, the growth of an individual, repeats phylogeny, the evolutionary history of that individual's ancestors. As a human embryo grows, it physically transitions through stages that resemble fish, amphibians, and reptiles, before finally taking on its mammalian form. In this 9-month span, the embryo undergoes a sequence of transformations that reflects billions of years of vertebrate evolution. If we follow these identical embryos to full term, they eventually diverge into the specialized adult forms we recognize. By tracing these lineages backward through the fossil record, we can see these separate branches of the evolutionary tree begin to converge. At the base of the tree, the lineages of fish, amphibians, reptiles, and mammals meet at a single point, a common ancestor. Diverse environmental pressures forced this single lineage to branch out, leading to the vast array of vertebrate life on Earth today. This embryonic evidence shows that vertebrates did not emerge as isolated groups, but as divergent branches of the same ancient family. For students in Class 10 Life Science, understanding these embryonic stages is key to explaining the evidence for evolution. To provide a complete answer on an exam, cite Haeckel's law and list the 3 structural proofs: the gill slits, the tail with its myotome muscles, and the 2-chambered heart. The fossil record provides a physical timeline of life's history etched into stone. However, an equally detailed record of our origins is preserved within the embryo, replaying our evolutionary story during the first days of existence.